Good morning to all lecturers and students. We are from Group 3. We'll be presenting Karyotyping on Trisomy 21. This is our group members. Table of content for this presentation. Karyotyping is done to identify and evaluate size, shape, and number of chromosomes in a sample of body cells, which then displayed as systematized arrangement of chromosomes pairs known as karyotypes. Karyotyping helps to determine abnormalities from adult chromosomes defects in women and in fetus, cause of baby's birth defects, and identifying sex of a person by determining presence of Y chromosome.
The pictures show the difference between normal and abnormal karyotyping of male individual. Extra copy of chromosome 21 can be observed, which cause the individual to have trisomy 21 with 47 chromosomes and XY karyotype. John Ladendown first identified Down syndrome in 1866, which links to chromosome 21, and discovered century later by Dr. Jerome Lejeune. It is defined as chromosome abnormality in which a child is born with an extra chromosome, and known as most common chromosome abnormality in humans. In Down syndrome babies, chromosome 21 has an extra copy, and trisomy 21 is another name for Down syndrome.
Trisomy 21 is caused by non-disjunction of chromosome 21 during meiosis 1 or meiosis 2. As a result, an embryo with non-disjunction has 3 rather than 2 copies of chromosome 21. A pair of chromosome 21 in either the sperm or egg fails to separate before or during fertilization. The additional chromosome is duplicated in all of the body cell as the embryo grows. Maternal age is linked to an increased chance of having a baby with trisomy 21. Some physical symptoms of Down syndrome may affect the ears, eyes, mouth, nasal bridge, neck, hands, fingers, and joint. Majority of people with Down syndrome will have some form of mild to moderate development delay that includes having poor judgment, impulsive behavior, short attention span, and slow learning capabilities. Down syndrome child might suffer a lot of health problems. Majority of Down syndrome babies will develop congenital heart defects, which develop during pregnancy. Most common congenital heart defect that occurs among babies is an atrioventricular septal defect.
There are 2 screening tests that can be done before the physicians come out with the diagnosis. First is blood test to measure pregnancy-associated plasma protein A and human chorionic gonadotropin (HCG). Moreover, nuchal translucency test can also be done, which is to provide information regarding possible birth defect in fetus by measuring the thickness of fluid buildup at the back of baby's neck. Next, amniocentesis can be done by inserting the needle through abdomen under ultrasound guidance to remove the amniotic fluid. The amniotic fluid will then be used as a sample to perform cell culture and karyotype analysis. This procedure can be done between 15 to 20 weeks of pregnancy. Lastly, chorionic villus sampling, which is removal of small samples of cells from placenta. It can be done by 2 ways, which are transcervical and transabdominal. This test can be performed between week 11 to 14 of pregnancy. Down syndrome cannot be cured, but early treatment programs can help to improve skills. There are a wide range of physical and developmental therapies available, including physical therapy, occupational therapy, speech therapy, and behavioral therapy. Other than that, programs like social and recreational activities can help to enhance the individual cognitive abilities. That is all from our group regarding the trisomy 21 karyotype.
Next is cytology lab staining. Table of content consists of specimen and technique used in this experiment.
For vaginal or uterine smear, we are using pulp stain to stain the cells. Then we observe the stain slide under the microscope for theta magnification. A nucleated epithelial cell without any presence of neutrophil was observed in the picture at the left. So we suggest the estrous stage for this rat is pro-estrous. We are comparing our finding with the reference image at the right side, and we can see an abundance of nucleated epithelial cell without any presence of neutrophils.
Next, Barker sample can be obtained by using swabbing technique. Under perpendicular stain, intermediate squamous cell as in the red circle and non-keratinized superficial cell as in the blue circle can be seen. This observation can be compared with reference image in the right side. The troubleshooting is the slide need to be observed under 100 magnification to get more clearer image.
For urine, we use voided urine as the specimen. Then smearing technique and Papa Nicolau staining were done to observe the urothelial cell under the microscope. Large isolated cells but unclear morphology of urothelial cells were seen. This may be due to improper smearing and staining technique. For reference, large flat polygonal cells and abundant cytoplasm can be seen for PEP stain specimen.
Dabbing technique is used to collect the cytology cell sampling from the rat kidney. MGG staining was used and small round nuclei was observed. Troubleshooting is done to ensure that the slides are air-dried properly to avoid artifacts. As for the human reference, dispersed cell with small and round nuclei is observed and normal nucleus to cytoplasm ratio is seen.
FNAC was used to collect cytology sampling of rat liver, followed by MGG staining of the cells, which showed presence of hepatocytes. Troubleshooting is required by applying proper technique for a better observation. As reference, human liver cytology observation showed presence of big purple cells representing hepatocyte stained with MGG.
Squashing technique was used to observe spleen cytomorphology. By using MGG stain, the presence of erythroid precursor and white blood cells can be seen. Troubleshooting need to be done, which use higher 100 times magnification to get the better view morphology of cells. As references, morphology shows erythroid precursors, band-aid segmented neutrophils, and lymphoid cells.
Dabbing technique is also used to observe heart tissue. A cell with distorted shape and without a nucleus is observed. We suspect that it is cardiomyocyte. The staining we use is May Grommet-Jimsa. However, only one cell is being observed in the field. We should repeat the technique or use a different technique to observe the tissue. As a reference, we have obtained a picture with H&E staining. The cardiomyocyte is rod-shaped with single nucleus per cell.
Dabbing technique was used to observe lung cytomorphology, wherein the stained red cell using MGG shows presence of columnar cell with oval nuclei. Recommended troubleshooting is do not air dry the slides for too long before staining. For human reference of lung cytomorphology, shows ciliated columnar cell, oval nuclei, placed at basal, and finely textured chromatin.
Lastly, for the scraping technique, we use it on the rat trachea. Picture on the left show our group result. The cell present is columnar, have oval nuclei with steeple chromatin. Compared to the reference, the cilia are not clearly seen. The staining we use is MGG. For troubleshooting, need to spread the sample thinly for better visualize.
That's all from us. Thank you.